Hi, Stacy.
Hi.
My connection kept dropping. I haven't had any problems at home. And this morning, the internet cut out twice on me.
Oh, really? At your house or there at the office?
No, at my house. So I just raced here.
Oh my gosh, did you really?
Yeah.
Well, I have been having a hard time connecting to my computer remotely today. I don't know if there's something going on as far as that's concerned, but I've been having to work on webmail instead of connecting to my computer. So I'm going to have to come there later and figure out what's going on. But I appreciate you racing there for this.
Well, I could just imagine we're halfway through and all of a sudden I lose internet at home and everyone's
Right.
wondering where'd he go?
Right. Well, we're recording right now just because I haven't asked you, but I would like to be able to post this on YouTube just for those folks who aren't able to participate today, if you're okay with that.
Yeah, I'm okay with that.
Okay. Are you able to share your screen or do I need to do something?
It says host disabled participant.
Huh. Yeah, it may be that you're not, because you're not logged into the Tamu Zoom system. Maybe that's the problem. So it doesn't know who you are.
Let me go.
You should be able to now. I just hit allow.
Yeah, I can now.
Okay, good. Great.
Did you see it?
Yep.
Yeah, full screen.
Yeah, that's good.
Okay.
Okay, I'm gonna keep working and keep paying attention to the time. The last time I did one of these, we had 15 people register and we had 5 people show up. So we'll see how it goes today.
Okay.
But I'm sure we'll have a few. So I sent them a reminder this morning, so hopefully that will help.
Okay.
Okay. Okay, it is 11:01, so we're going to get started. I'm sure a few more people will join us. Hi, everybody. It's good to see your names at least, and maybe some pictures. Thanks for being here today. We're trying to provide some professional development to you while you're at home and working. And so Dr. Brett Mitchell is here today. He has offered to give this presentation. This is one of the subjects that those who filled out the postdoc survey several weeks ago mentioned that they would like to see some information on. So he's going to give us a presentation on effective scientific presentations. We're expecting about 17 people today. And so I think we are going to keep this casual and allow you guys to ask questions, but I think just to make it easier, if you do have a question, just put it in the chat and then I will vocalize that at the end, and Dr. Mitchell will answer those questions if that's okay with everyone. One reminder, so 2 weeks from today, same time, 11 o'clock, we're doing a webinar on writing an excellent review article with Dr. Barbara Gastel. Email me if you haven't seen the communication about that, and I can send you the link to RSVP for that. And then 2 weeks from then, Barbara Gastel will also do another webinar for us. So keep your eyes out on your email for stuff like that. So just real quick, we have Dr. Brett Mitchell here today. He is an associate professor in the Department of Medical Physiology in the College of Medicine. He's also the director of our summer undergraduate research program here in the College of Medicine, which I believe was canceled this summer. Is that right? Yeah, it's too bad. It's a great program for our undergraduates to get to know our faculty and our college and get a lot of research experience. And I'm sure a lot of y'all have worked with those students over the years. Dr. Mitchell has a BA in health fitness and biology. He also has an MS in human physiology and a PhD in physiology. He did his postdoc at University Space Research Association at NASA's Johnson Space Center in Houston. And please welcome Dr. Mitchell.
Thank you, Stacy. Pleasure to be here. This is something I do informally for our summer research program participant. So I basically call it how to give a 10-minute talk, but it really can be applied to whether it's an hour seminar that you're invited to give somewhere, or maybe even a future job interview for you postdocs. You know, when you go somewhere, you're going to have to give a seminar. It's typically 45, 50 minutes long, but for our purposes today, this scenario is really kind of a 10-minute talk format. So all of you submit an abstract to a national conference. An email back saying it's been accepted, and whether it's virtual or in person, you go to the meeting to present your 10-minute talk. All right. And over 20 years of attending conferences and moderating sessions at these national conferences, I've seen a lot of really good things and I've seen a lot of really bad things. So what I'm here today to tell you about is kind of the pitfalls and, you know, the bad things that I've seen, and then what I would highly recommend from presentations that I've seen that are really, really good. And ideally, you want people coming away from those conferences saying, wow, so-and-so from Texas A&M just gave a really great talk. And I remember the take-home message. And you know what? Down the road, I have seen people be selected to give seminars or attend conferences based on past presentations that were well received. So this is something that's going to follow you throughout your scientific career. I've seen some really big names in a variety of fields where they get up and everyone in the audience is just stoked to hear them talk. And it's just a boring presentation and data slide, data slide, data slide. You walk away from that thinking, wow, they're a great scientist, but they really didn't present their stuff very well. So the purpose of today is to get you kind of prepared and you know, when you're thinking about giving your talk, kind of prepare ahead of time, some tips while you're giving your talk to focus on, and hopefully everyone goes away thinking, wow, that was a really great presentation. I heard so-and-so from A&M talk several years ago, and hey, let's get them on the program because they are such a good speaker. So, so hopefully some of these tips will sink in. I'm not trying to offend anyone. I do some of these a lot of the time as well. So again, these are just things that I've seen and I try and convey. All right, so again, the scenario is a 10-minute talk, and we're gonna kind of walk through what that looks like. So tip number one is to prepare. All right, you've submitted your abstract, you've got it accepted. They say it's gonna be an oral format, 10-minute talk, a few minutes of questions. All right, so what you need to do is organize your slides to tell a story. All right, so just throwing up data slide after data slide is not going to be helpful. All right, you want people to leave there going, wow, that was a really neat story. All right, and one way to tell if that's effective is to time it out and practice. All right, so if you're practicing and you're consistently 14, 15 minutes long, those are not well organized slides. You're trying to fit too much in amount of time. So really, it's okay to cut out sections, or you don't have to throw everything from your project into that talk, especially on a 10-minute talk. So time it out, practice it, all right? So you don't want to go to the meeting, well, you know, perfectly rehearsed word for word, okay? But you should know the order of the slides, all right? So, and you'll see as we do a little bit later. So we're going to go through some tips, and then I'm going to give a kind of abbreviated 10-minute talk of my research, and I'm going to do some things maybe subtle, maybe not so subtle of what we're discussing. And then hopefully we'll have time where I can do like a real one where we address some of these. But back to, so know the order of the slides. Again, don't have to have the words memorized for every slide, but know which slides are in order so that you can have smooth transitions and you know the flow of your slides in your presentation. Have an accessible backup file. So I have seen people get to the meeting, and usually the day before you give the talk, you have to go into a speaker ready room or a presenter ready room, and you will have to upload your slides. And I've seen too many people put their flash drive in and all of a sudden it's not formatted right, or it doesn't work, or got damaged in the trip or something. All right, so have an accessible backup file, whether that's, you know, email it to yourself that you could pull up on the computer and access that file, and do a cloud backup that you could access from a web anywhere. So have an accessible backup file when you go to that meeting. All right, so you're at the meeting, all right, so your session's coming up. Usually there's 15 to 30 minute break between sessions, all right? So say your session is at 3 o'clock in the afternoon. Well, the session before ended at 2:30. All right, people start to trail out. Some are going to stick around and talk, but your job is to get in there shortly after 2:30 and go find the moderators. All right, they may or may not be there yet, but go up, if they are there, introduce yourself to them, look them up so you know what they look like before you get to the meeting. Find them, introduce yourself. If you have a difficult name to pronounce, help them with the pronunciation. It's a little embarrassing when they get it, if you haven't met them or talked to them and they get up and just butcher your name, and then you have to correct them. So that it's okay that happens, but if you can kind of help them with pronunciation ahead of time, they really appreciate that. So go up and introduce yourself, let them know that you're there. All right. As a moderator, it's really nerve-wracking when I'm not sure if all the speakers for my session are there or not. All of a sudden you see someone walk in late and you hope that that's them. All right, so introduce yourself, let them know you're there. Get familiar with the tech and the visuals. So get up on the stage or the podium, learn where everything is. All right. It's really awkward when someone gets up there and 10-minute talk. They're starting, they get up there to give their real talk and they don't know how to advance the slides. They don't know whether it's a laser pointer or the mouse. They don't know where to look. They don't know what the lights on the podium mean. So get up there and get familiar with it. So it's kind of like being in the cockpit of a plane. Get to know where everything is. There's usually a tech person there that will help you get your slides loaded and make sure they work. All right, that's the next one. So are you going to flip through with the arrow button on the computer? Is it a handheld remote? Learn what buttons do what, okay? Are you going to use a laser pointer where you have to look up at the slides, or is there a TV down below? And that's where this visuals come in. All right, so bigger meetings, bigger rooms are going to have a TV down on the floor in front of you. Are you going to look at that? Or are you going to have to turn your head and look at the screen next to you? So just kind of understand what that looks like. All right. Ask, is it going to be a microphone on the podium, or is it going to be a lapel mic that you're going to have to clip on? That makes a difference as to how you stand and where you look and so on. So really get in there and get comfortable. So what I've seen a lot of times, again, people show up late, they're not comfortable with that. So the moderator has to stand up and tell them how to advance the slides, and they're just wasting the audience's time at that point. All right, so really get in there and learn where everything is. I had one instance where there was a podium for a time check where it went from red to yellow to, or green to yellow to red in terms of their timing. Well, this speaker showed up really late. I didn't even know if he was coming to the session, called his name, he came up, and when the light changed from green to yellow, he sped up and raced through the rest of his slides, and it turned out he had about 4 minutes left. All right, so people are walking away from that presentation like, ooh, that wasn't very good, it was rushed and everything. So again, get familiar with the podium, learn how to operate everything. Again, make sure your slides work, especially if you have videos in them, make sure that you can see them. And if there's time before the session starts, go ahead and meet the audience. So some of those people in the front row, go up and introduce yourself, ask where they're from or something. Because once you get to know them a little bit, talking to friends makes you feel a little more comfortable, okay? So if you're just kind of hiding in the corner until it's your turn and you get up there and you think, everyone out there is a stranger and these are all the experts in the field. Well, if you've already met a couple of them, you kind of puts you at ease a little bit. All right, so we have prepare and arrive early. Now we have a smooth start. So it's your turn to start talking, all right? So the moderator introduces you, you're so-and-so from Texas A&M. And sometimes they'll give the title and sometimes they won't. But the first thing to do is thank the moderator with humility. I've unfortunately have seen some very, very big names get up there and say, wow, you're really lucky to have me here. I'm so busy. I was able to fit this into my schedule. I've heard some very arrogant things, and it just doesn't start the talk off very well. So thank the moderators with humility. Say thank you for having us. Thank you for selecting our abstract. I've also seen this, where if the moderator says it's so-and-so from Texas A&M, and they rattle off a 28-word title, and they need oxygen halfway through it, all right, if you get up there and start your talk and say, thank you, so-and-so moderators for selecting our abstract, the title on my talk is, and you start repeating it, everyone in the audience just visibly goes, ugh, All right, you're just wasting a minute out of your 10 minutes. All right. So if the moderators says the title of your talk, just thank the moderators. Again, if you know them by name, go ahead and thank them by name. If you don't, just say thank you for this opportunity. Again, repeat the title, don't repeat the title. Definitely do not repeat the title. All right. So if they say the title, just say thank you for this opportunity. I'm excited to talk to you today about our work that we've recently produced in the area of hypertension in my case. All right. So that tells the audience that, okay, this is what this is going to be about. And then you have to provide any disclosures. And that's usually on the first or second slide. And please adhere to that. So I have seen a talk where there were no disclosures at the beginning. It was all about a GSK drug. Everything was, this drug was beneficial. It prevented everything. It was great. And then at the end, it's like, oh, this person collaborated with GSK who paid for the whole study. So the audience needs to know that up front. All right. So again, this is a smooth start. This is you're just starting out. Thank the moderator and kind of get you started. All right. So tip number 4 is get into a good flow. So the first couple slides is really where you're already nervous. That's okay. Everybody gets nervous. All right. But if you get into a good flow, you'll start to feel more comfortable and it'll go real smooth. All right. So the first thing to do is to speak clearly. All right. If there's any runners out there, as soon as a gun goes off in a running race, what does everyone do? They start off way too fast. All right. So same with public speaking and effective presentation. Speak clearly. All right. If you start speaking too fast, you're going to start speaking fast the whole time. All right. So start slow, speak clearly. All right. Sometimes at the beginning, a little humor in some cases is okay to help get comfortable. All right. Having a little cartoon or, verbiage or a saying or a quote or something, if you see the audience give a little chuckle, that kind of makes you feel comfortable and the audience is like, okay, this is interesting. I'm into this. All right. So there are some cases, don't put up offensive cartoons, or if you're talking about something really grim, make a joke out of it or something. So kind of assess your situation and if it's appropriate, it's okay. Don't read all the bullet points verbatim. They've done studies on this, and basically by the time you get to your second bullet point, the audience has already read the whole slide, okay? So if you get to the second bullet point and you're reading it word for word, all right, the audience has already read it. So if you get to the third bullet point and you're reading it word for word, the audience initially says, okay, this person's gonna read to me like I'm a 5-year-old, all right? So mix up your verbiage, all right? Don't read it word for word. Focus on the awake audience members. So this is not to offend you that everyone will go to sleep, all right? But I have seen a lot of presentations, okay, where there will definitely be some people in the audience who are distracted. They're on their laptop or on their phone. I have had audience members that are literally asleep while I'm talking. National conferences, people are walking out the door and people are coming in the door in the back during your talk. That will happen, okay? I have seen many people, especially trainees, when they're giving their 10-minute talk at a national conference, get distracted by that. All right, they start focusing on who's coming in, who's coming out. As a trainee, when I was giving a talk, I would look up at the person sleeping. And every time I looked up to make eye contact with the audience, he was the first person I looked at to see if he was awake. And every time I looked up, he was still asleep. And in my mind, I'm thinking, wow, I'm not doing a really good job. That person is literally asleep. All right. So find the people that are interested, find the people on the edge of their seat whose eyes are wide open and they're really into it. Okay. So when you look up to make eye contact, talk to those people. Don't focus on the negatives and the people coming and going, the people talking in the background, or people looking down at their device. All right. Because that in your mind says, wow, I'm not doing a really good job. All right. You can't control that situation. What you can control is talking to the people that are really interested in your work. All right. Find that person whose head is nodding when you're going through your talk. Okay. Then when you look up, talk to those people. All right. So go from their eyes to the next one and to the next one. All right. And then when they leave, they were obviously interested in your topic. They were obviously interested in your presentation. All right. And when they leave, they're going to be like, wow, that was a really great talk. It was almost like they were talking directly to me. All right. So stay positive and concentrate on talking to those people, and that will help you get into a good flow. All right. Tip number 5, explain the figures well. Okay. So just throwing up figure after figure it's not really helpful. The audience, it doesn't understand. All right. So I kind of have these 4 things for each figure that you put into your presentation. Why you did the experiment, what you did, what you found, and what it means. So each figure, you should be able to explain those 4 things, okay? So why you did the experiment, okay? In order to see if blood pressure went up, we did this, all right? So what we did was, okay, we gave the mice this, and then we measured blood pressure, all right? And what we found was, okay, blood pressure went up in the experimental group, and this means that this stimuli that we gave this group caused high blood pressure, all right? So all of that took about 20 to 30 seconds, okay? So if you do that for each figure, especially in a 10-minute talk, you can tell you're not going to have a ton of figures. So trying to fit figures in without going through these was really going to confuse the audience, okay? So explaining the figures, why you did the experiment, what you did, what you found, and what it means. All right, the next tip is explain the data well. All right, so explain the figure well, but also explain the data well. So when you show a figure, go through what was measured. All right, explain the axes and the units. On the x-axis, you'll see time. On the y-axis, you'll see blood pressure. All right, explain the control group or the normal group. So if you just throw up a bunch of, and I've seen this before, it was just hit immunofluorescence after immunofluorescence after, immunohistochemistry after immunohistochemistry, and they never explained what we're looking at. So, are we looking for the brown staining? Are we looking for the red staining? Are we looking for the black staining? They never explained the control so that the audience really couldn't see what the difference was. All right. So by fully explaining the control group, then you can show the experimental group and how it differs from the control. All right. So again, explaining the data well. What was measured, explain the axes and units, explain the control or normal group, and then explain how the experimental group is different. All right. So wrap it up cleanly is tip number 7. So you've gone through all your results. Now you get to your summary, conclusion, or future direction. All right. It's personal style, whether you have 1, 2, or all 3 of those. But you don't want to have your out of your 10-minute talk, you don't want this to be the last 4 minutes of your talk. So wrap it up cleanly, have concise bullet points, because this is almost the last thing they're going to walk out of from your talk. Okay, this is what they're going to remember. So this is your place to kind of put it in their head that when they leave, this was the take-home message. All right. Keep the people acknowledgement concise. So you don't need to thank your 3rd grade teacher who got you excited about science. Okay. Really acknowledge the people who contributed to the work, collaborators, and so on. And also with funding, be sure and have a concise acknowledgement. So a lot of science, most of science can't be done without funding. Be sure you acknowledge that as well. And at the very end, say thank you. All right. So I've seen a lot of times, and this happens all the time, you think you're done and then you say, any questions? And as a moderator, that just kills me, because some people start clapping, some people start walking to the microphone, some people start raising their hand. All right. So the best thing to do at the end of your 10-minute talk or presentation is to say thank you. That clearly indicates that you're done. Then the audience can clap, and then the moderator can say, okay, this is open for questions. The speaker will take questions now. This abstract is open for discussion. So let the moderator do their job. And by you saying thank you, that shows a clear sign that you're done. All right. So that's how you wrap it up cleanly. And you think you're all done, right? You've given your 10-minute talk. You just feel a load off. You're like, all right, you take a breath. Oh, and now you've got questions from the audience. Then you get even more nervous than you were before this whole talk even started. All right. Now the experts are walking up to the microphone, and I see this at every meeting. All right, so the number one expert's going to get up to the microphone. You're nervous about the experts standing up. You're nervous to handle questions. They're going to make me look stupid. All right. And the first person gets up and says, I have a 3-part question, and runs through about 2 minutes of questions. And you know what the first thing the speaker says is, can you repeat the question? And the whole audience just visibly goes, all right. So a good way to handle questions, and especially the first couple after you've given your talk, is to repeat the question back to them. So I have seen this a number of times, is where you get so flustered to handle the question that you really do know the answer. It's just you're so flustered you can't grab it from your brain. So a good way to buy some time is repeat the question back to the audience person. All right. Let me understand if you're asking this, okay, and then repeat the question back to them. Or am I correct in saying that you're interested in how we did this Western blot? All right. That gives you a little bit of time. It helps you digest the question into more understandable terms. You're buying time to dig in your memory bank. Okay. Oh yeah, that's really not that bad of a question. I know the answer, and then you can give them the answer. So that's a good way to buy some time and digest it. If you keep asking them to repeat the question, and I've seen this, 6 questions were asked, and every time it was, can you repeat the question? All right, that's a little frustrating for the audience and moderators and so on. All right, and it really kind of, the audience is taken away from your presentation, is that you're not really listening, or you're not thinking. So you don't want to leave your 10-minute talk with that in their mouth. Someone will inevitably stand up and say, have you measured, insert their gene or protein of interest? All right. So you're doing a study, and I study IL-6. I'm going to stand up and say, have you looked at IL-6 in your model? Don't say something like, that's a stupid idea, please sit down. Okay, I've seen something like that. The best thing to do is to say, thank you for the suggestion. We have not measured that, but we will consider that moving forward. That's the best way to handle those questions. But every talk will get that question, have you measured this? All right, so just handle that. Don't say, no, we haven't measured that. I'm not sure why we would measure that. So don't make the person asking the question feel stupid. Just say, thank you for the suggestion. Make them feel smart, like they're helping you with your research. All right. I've seen it all, trust me. Another way to handle question well is it's okay to say if you don't know, but you'll check on it and get back to them. That is a legitimate answer, okay? So don't be afraid. Don't start speculating too much. Don't lie. It's okay to say, you know what, I really don't know offhand, but I would like to check on that and get back to you, okay? And then lastly, this inevitably happens a couple times, is you get some person asking questions who just won't relent and keeps bringing up, and it turns into a one-on-one discussion, all right? The audience really isn't interested in that. A lot of times the moderator will say, especially if you're running out of time, say, can you two continue this conversation outside after the session? It's okay for you to say, especially if you think it's gonna be, you really have a long defense or a long answer, it is definitely okay to say, I would love to talk after this session with you. Okay, so that's a good way to kind of handle that question and answer session, which can usually be the most nerve-wracking. But again, digesting the questions so that you understand them really gives you some time to provide good answers. And trust me, when people leave sessions, and the trainees especially are able to handle questions for more experienced people in the field, that is a real confidence boost for them. And the audience leaves like, wow, they really know their stuff. Okay. So, all right. So to summarize, these are the 8 tips for effective scientific presentations. Number 1, prepare. So have your slides organized, time it out and practice. All right. I have had, as a moderator, people starting to go 12, 13, 14 minutes, and they're still in their results. All right. Those are people where I have to interrupt them and say, you need to wrap it up. All right. Now, if they're 10 or 11 minutes and they're on their acknowledgments, it's fine. All right. But if there's no end in sight and you're going long, what that does is put the moderator in an awkward position, because they're going to have to interrupt you or even end you, right? It puts the session behind. So everyone's, people trying to run in to watch the next talk. Well, you're running late and that messes up everyone's schedule. So really be sure you prepare, time out your talk and be around 10 minutes. Arrive early. Make sure you're comfortable with everything. Make sure you know how to advance slides. Make sure you know where to look. A lot of times, and I might mimic this, but there'll be a podium mic, all right? And when the person looks away at the screen, they get real silent. And then they come back and they get real loud again, all right? So when they look away and get real silent, everyone in the audience says, what are they saying? What am I missing? What are they saying? And then they come back real loud and say, oh, I just missed what they were trying to say, all right? So arrive early, get comfortable with the scenario, get comfortable up on the stage, kind of figure out where you're gonna look and practice it, okay? Get off to a smooth start, speak clearly, speak slowly, don't read word for word. It's okay a little bit at the start just to get you comfortable, but really you want to kind of start getting the audience involved. Get into a good flow, all right? Explain the figures well, explain why you did the experiment, what you did, what you found and what it means. Explain the data well. So don't put up figures that you're not going to explain. All right, I've seen that as well. People will put up an 8-figure panel and they go through 2 of them and then they go to the next slide. So if you're going to put it up there, be sure you explain the data and explain it well. Wrap it up cleanly. So have a summary or conclusion, concise acknowledgements, and thank you. Always say thank you at the end. And then handle questions well. So we went through that. So how are we doing on time? All right. Stacy, how are we on time?
We're good. It's only 11:30 and we blocked off an hour for this. So
okay.
If you have other tips or if you want to take questions, whatever you want to do.
What I'd like to do is do an abbreviated 10-minute talk. And each slide, I'm going to try and do something that we just went over of what not to do. Okay. Just so you can kind of see and catch, some will be subtle and some will be not subtle. Okay. So, and this is my research and I don't have a lot of data. So it's just something that we can use to go over. All right. So I'm at a national meeting. Our abstract was selected for a 10-minute talk. The moderator just said, and from Texas A&M, our next speaker is Brett Mitchell. The title of his talk is Clip Knockout Mice Are Resistant to Toll-like Receptor-Induced Preeclampsia. All right, I get up and say, thank you, moderators, for having us. So the audience is really lucky that we're here. Title of my talk is Clip Knockout Mice Are Resistant to Toll-like Receptor-Induced Preeclampsia. Preeclampsia, or PE, is the de novo development of hypertension and proteinuria end organ damage. Preeclampsia may lead to seizures, eclampsia. It affects about 6 to 8% of all pregnancies, incidences rising. Leading cause of premature births, associated with CV cardiovascular disease later in life. There are no effective treatments currently. And I put this cartoon in because when I read it, I just died laughing. Right. The dangers of pregnancy. So we think the innate immune response is involved in the development of preeclampsia. We think that there's pre-existing endothelial dysfunction, such as obesity or advanced age, plus pregnancy initiates an innate immune response, which leads to inflammation, endothelial dysfunction, and preeclampsia. We also believe that there's placental dysfunction, so that if there's an abnormal pregnancy and fetal rejection, there's an innate immune response leading to preeclampsia. So the innate immune response is also involved where we think it's just highly active, all right? And then you throw on a normal pregnancy and that can lead to preeclampsia because of the excessive immune response. And this is evidence with regard to infections as well as autoimmune disease. So we know that women with a history of autoimmune disease such as lupus are at higher risk of developing preeclampsia. So the danger receptors in our body are toll-like receptors. All right, these are on the outside of immune cells as well as other cells and inside the cell as well. And activation of these leads to inflammation. Our group and others have shown that during pregnancy in animals, if we activate these toll-like receptors, we can induce a preeclampsia-like syndrome. So how does TLR activation cause innate immune cells to become pro-inflammatory and contribute to PE pathogenesis? So we think that that CLIP expressed in MHC class 2 on immune cells is involved. So we know that innate immune cell, when its TLR is activated by either TLR3 or TLR7, that CLIP is expressed in the groove of MHC class 2 and blood pressure goes up, protein excretion goes up, fetal demise goes up, and vasodilation goes down. So what happens if there is no CLIP? Would this immune cell then die? So our hypothesis is right here, as you can see. So we had pregnant and CLIP knockout mice. We injected on these days and we sacked on this day, and we did these measures, as you can see here. So it's pretty obvious in this picture that they did not develop hypertension. The CLIP knockouts did not develop hypertension. They also did not develop endothelial dysfunction, which is pretty obvious on the right panel. What's this slide? Oh yeah, urinary protein did not go up in the CLIP knockouts. Oh yeah, this is we measured cytokines. And this is the pregnant mice get an increase in pro-inflammatory cytokines. Oh, I forgot we even put this in here. And the CLIP knockout mice didn't go up. So our conclusion is that CLIP knockout mice don't get preeclampsia. I want to acknowledge Valerie. Valerie is great. She's so much fun to have in the lab. She works really long hours, works really, really hard and contribute a lot to this project. Dr. Chatterjee, she's really quiet, but she's a really good worker. Again, really quiet, but sometimes she can be so funny. The one time I was drinking water and she said something just really funny about science and just water came out of my nose. She was so funny. So, but usually pretty quiet. And Kelsey is pretty new to the lab. She didn't do a whole lot on this project, but we're hoping that she starts contributing more. I'd like to thank these other people and our funding. Are there any questions? All right, so hopefully you caught on some things that you should not do, right? So I'm gonna go through them kind of real quick. How many people do we have on, Stacy? 18, 16?
I think 15, okay. Not including us.
All right. Can anyone chime in on what I did wrong on this slide?
You mentioned that we were lucky to have you here.
Yep. So did not show humility, showed a little arrogance. Thank you, Alex. There was one other I did on here.
Reading the title.
I repeated the title, right? Even though it's not a 28-word title, it's still the audience is like, we just heard that. All right. Oh, and I didn't reveal my disclosures even though it's listed. Okay, so that's what I did wrong on that one. What did I do wrong on this one? I did two things primarily wrong.
Read the bullet points.
I read it word for word. Good. How about the cartoon thing? Was that necessary? I didn't need to make a big joke about it. I think by itself, it would just kind of be on its own. One thing we didn't talk about that I did on this. Did anyone catch it?
Mouse was really distracting.
Yeah, exactly. Thank you. And this happens, some people get nervous, especially the days of the laser pointer, when you're trying to highlight something and they're constantly circling it, underlining it, and the whole thing. So good catch. So I was constantly using the mouse, and that's very distracting. So yes, I'm trying to point out things, and really what you should do is go, okay, here, and then leave it alone. Or with the laser pointer, just kind of point, underline, and then leave it alone. The idea is to get their eyes to that area on a busy slide like this. But you don't want to be distracting them with constant movement. Another thing related to that is having PowerPoints that have a lot of animation. That's a personal style thing. But there's good presentations with animation and there's bad presentations with animation. So just keep that in mind. Sometimes it can be excessive and distracting. So keep that in mind. I didn't explain this fully, and I tended to use the mouse a lot. Was the main thing I was trying to do there. I didn't explain those fully as well. Anything here? I said, here's our hypothesis, right? And didn't read it. This is the one area where it's not too bad to go ahead and read word for word. This instills in the audience, this is what we're testing. Anything on this? Was this well explained? Did you understand this slide?
No.
Okay. So I didn't do terrible on this one. I didn't explain it from left to right though. So a good way to explain this is start from the left, the normal, then proceed from left to right. So most slides we're going from top to bottom, left to right. Okay, so starting off with clip in this immune cell, all right, I really should have gone over and say, okay, this is what happens at baseline, and then here's what happens after activation. All right, so hypothesis, again, this is okay to read. Oh, this is why I said this is our hypothesis, as you can see. All right. So read your hypothesis, that's okay. What did I do wrong here? Anyone? Kind of two things I did wrong.
Yeah,
And you,
the experimental. Oh, sorry. Go ahead.
Go, Shobana.
Yeah, I think you didn't explain the experimental design. I mean, in detail. Like, why did you choose the day 13, 15, and everything, and the results also?
Very good. So I didn't explain this design, what these are, and so on. And then, of course, right? So I just showed picture and said we did this. So I didn't walk through what these represented or what methods these measured and so on. So very good. What did I do wrong here? So two things. Two of our major tips that I didn't do here. One, I didn't explain the figure well, right? So I didn't say what we did well or why we did the experiment, what we did, what we found, and what it means. Right? I didn't explain the data well. I didn't explain what this measure is. I didn't explain the axes. I didn't explain the control groups. I didn't explain the experimental groups. Okay. So I did that here as well. So you see the difference? So just saying they didn't develop hypertension, well, that's my take-home message, but you don't get that from the slide. You don't even know what groups are, what, you don't know if that's good or bad. So same thing with this. What did I do wrong here? I did not transition into this next slide, right? So a transition into here, I basically just threw it up and then looked at what it was and then gave you the take-home message. Same thing with this, and I see that all the time. People who don't practice, they go to the next slide and then they have to read the title to remember what it was, all right? I've seen people look at the labels just to see what it was, okay? So again, know what's next, know what's coming, transition into it, explain it well, all right? And same thing with this. All right, so I could have gone through this a little bit more in depth, right? So this is where it was too short a conclusion, and I really didn't give a take-home message or a future direction. All right, what did I do wrong here? Well, this one was pretty obvious, right? So don't talk about personal stuff when you're acknowledging people. Keep it professional. What did they contribute to the project? Don't need to spend a lot of time, as I said earlier on. Again, inside jokes, the audience isn't going to know. So don't air grievances in the acknowledgement section, obviously, but just thank the people that contributed to the project. Again, you don't have to list everyone if they had minor roles or if they were in the lab at the time, even though they're listed, but highlight the people who contributed. And be sure and you highlight the funding agencies. I skipped over that. All right, and then what did I do wrong here? I said, are there any questions? All right. So we can either do a Q&A or I can go through this properly. Any preferences?
I think we have time to go through it. We have 15 minutes.
All right. So hopefully correcting everything that you caught. We'll hopefully do this in a better, more effective way. All right. So thank you, moderators, for selecting our abstract. I'm excited to present to you today about recent data regarding CLIP knockout mice and preeclampsia that's been generated from our lab. Here are our disclosures. So preeclampsia, or PE, is the de novo development of hypertension and proteinuria, which is protein in the urine, or end organ damage, such as dysfunction to the kidney, blood vessels, and so on. Preeclampsia may lead to seizures, and this is very serious, and that's called eclampsia. And if eclampsia occurs, the delivery of the baby is ensured at that moment, regardless of gestational age, and oftentimes leading to premature infants. Preeclampsia affects about 6 to 8% of all pregnancy, and unfortunately, the incidence is rising. The incidence is about doubled over the last 10 years due to a variety of factors. PE is the leading cause of premature births, and it's been shown recently that women that have preeclampsia, even though their blood pressure goes back to normal afterwards, are at 2 to 4 times higher risk of developing cardiovascular disease and stroke later in life. And right now, there are no current treatments. So the dangers of pregnancy, we believe the innate immune response is involved at several levels. All right, over here, we think that one method of development of preeclampsia is that there's pre-existing endothelial dysfunction. The endothelial dysfunction is evident in obesity as well as advanced age. So if you are entering pregnancy with dysfunction, you throw on the normal stress of pregnancy, the volume expansion and the stress. We believe that the innate immune response in some women leads to inflammation, endothelial dysfunction, and eventually preeclampsia. We also believe that there's fetal rejection. So if the pregnancy isn't proceeding normally, that the body, and what happens in nature is that the mom will reject the fetus, save herself so that she can try again. And we believe the innate immune response is involved in that. We also believe the innate immune response is just hyperactive in some women, that plus the normal pregnancy, that the body interprets that as dangerous, and that leads to inflammation and endothelial dysfunction and preeclampsia. And there is associative evidence that certain infections are associated with preeclampsia, and there is a high incidence of autoimmune disease in preeclampsia, such as lupus and others. So what is it in the body that senses this danger? Well, these are the toll-like receptors, and these are our body's defense system against pathogens or endogenous danger molecular patterns. So TLRs are located on the surface of cells or on endosomes inside of cells, but primarily innate immune cells as well as others. When these get activated, that leads through a signaling cascade, ultimately leading to inflammatory cytokine gene production as well as type 1 interferon genes. Right? And this is an antiviral response when TLR3 is activated as well as TLR7, which is relevant today because COVID acts on those TLRs. So in the past, our group and others have demonstrated that if we activate these TLRs with a viral mimetic for TLR3 or TLR7 and 9, that this fake virus can induce a preeclampsia-like syndrome by inducing inflammation. Others have shown that activation of TLR4, TLR1-2, and TLR2-6, which are the bacterial components or LPS sensors, can also lead to preeclampsia. So both bacterial and viral activators can lead to preeclampsia. So how does TLR activation cause the innate immune cells to become pro-inflammatory and contribute to preeclampsia? Well, we believe that TLR activation in an innate immune cell leads to this activation. So innate immune cells express MHC class II, which is inside the cell at normal baseline. And they also express CLIP. Okay, so when TLRs get activated, CLIP is expressed in the groove of MHC class II, and that is expressed on the surface of the immune cell. CLIP acts as a placeholder so that the immune cell stays alive and becomes pro-inflammatory, thus causing disease. Our previous research has demonstrated that this CLIP activation causes inflammation, or CLIP activation causes inflammation, and that leads to disease effects in a pregnant animal. And TLR activation can be induced, in our case, by the viral mimetics poly-IC, which acts on TLR3, or R837, which acts on TLR7. And the P mice act as our pregnant controls. So our question here was, what if there isn't CLIP that keeps this immune cell alive? Maybe this immune cell dies and all of these negative effects are reversed. So we hypothesized that if CLIP expression on innate immune cells plays a role in the development of preeclampsia, then mice that don't have CLIP will be resistant to developing experimental preeclampsia following TLR activation during pregnancy. So as I mentioned, we used pregnant mice as controls, as well as CLIP knockout mice. We gave them the TLR agonist, either the TLR3 agonist or the TLR7 agonist on days 13, 15, and 17. And this was to allow the animal to get pregnant, the placenta to form. Then we started on these days. And these agonists have a 48-hour window of activation. If we start too early, the mouse undergoes spontaneous abortion and blood pressure never goes up. So we injected on days 13, 15, 17, took blood pressures, and then euthanized the mice on day 18, in which we harvested the tissues. So here's how we measured tail cuff blood pressure in the mice. We also performed real-time PCR to measure changes in gene expression, as well as Western blots, flow cytometry, and vascular reactivity to measure endothelial dysfunction. So as you see here, so we wanted to see if the CLIP knockout mice did not develop hypertension. So we measured systolic blood pressure by tail cuff on days before the injection on days 13, 15, and 17, as well as in between days. So in wild type mice, a normal pregnant mouse, blood pressure is about 100 systolic kind of throughout pregnancy. But in the pregnant mice that are wild type, when we give the TLR agonist, they get hypertensive, as seen here, blood pressure is around 140. What was really interesting was that in the CLIP knockout mice, really no effect on the blood pressure in a normal pregnant CLIP knockout mouse. But when we gave the TLR agonist, there was no significant increase. So what this means is that the CLIP knockout mice did not develop the hypertension seen in preeclampsia. So next we wanted to see if there was endothelial dysfunction as we see in wild type mice given TLR agonists. So here we're measuring relaxation from phenylephrine. So we take the aorta out of the mouse, we put it in a myograph and trick it into thinking it's still in the body. It's oxygenated and warmed to 37 degrees. And when we give phenylephrine, which is basically adrenaline, the blood vessel contracts. Then we give increasing doses of acetylcholine, which is a vasodilator, and we measure how well the blood vessel can relax. So in a wild type, normal pregnant mouse, what we see is we see a contraction up to here. And then as we increase the dose of acetylcholine, we get a significant drop, which is vasodilation. In the hypertensive mice that were given the TLR agonist, there was a significant decrease in relaxation. So this is evident of endothelial dysfunction. So in a normal pregnant clip knockout, we see normal relaxation of the vessel after constriction. And in the TLR agonist mice that weren't hypertensive, their blood vessels were able to relax normally. So they did not develop the endothelial dysfunction. Next, we wanted to measure proteinuria, which is a characteristic of preeclampsia. So here we're measuring urinary protein as a concentration. So, and that urine was taken at euthanization on day 18 of pregnancy. So there's about 60 milligrams per deciliter in a wild type pregnant mouse. In the hypertensive TLR activated mice, there was a significant increase in the amount of protein in the urine. And this was reduced and was not significantly increased in the clip knockout mice given the TLR agonist. So proteinuria did not develop in the clip knockout mice. So next we wanted to see if there was evidence of inflammation. So we measured the percent of these pro-inflammatory cytokines, IL-17A, interferon gamma, and TNF alpha. And this was measured in the plasma after euthanization on day 18. And you see normal levels in the wild type pregnant mice. And we use this to normalize the levels in the plasma of the other groups. And what you see for all three pro-inflammatory cytokines was a significant increase in both the TLR3 agonist group as well as the TLR7 agonist group. So then we looked at these pro-inflammatory cytokines in the clip knockout mice. And as you can see, there's no significant increase of any of the three pro-inflammatory cytokines. Thus, inflammation was not induced in these groups. So under normal conditions, necrotic tissue or pathogens activate toll-like receptors. This activates CLIP it makes CLIP be expressed on MHC and activates the innate immune cells. There's no cell death, and there's an increase in pro-inflammatory cytokines. This leads to inflammation, endothelial dysfunction, and preeclampsia. So by knocking out CLIP, we were able to negate this effect, and we found, and the take-home message is that CLIP knockout mice prevented the development of TLR induced preeclampsia. Thus, CLIP inhibitors may be a potential therapeutic target in the future. I'd like to thank Valerie, Dr. Chatterjee, and Kelsey for their hard work on this project. I'd like to thank our collaborators and our funding agencies, as you can see here. Thank you. Then the moderator would say this is open for questions, and I would handle questions. All right. Any questions? We have a few more minutes. Could you tell the difference between could you tell the obvious differences between the first version and the second version? Okay. Let me pop up.
Dr. Mitchell,
Yes.
This is Kadris. I am Dr. from Sarabji Lab. Thanks for your presentation. Really, it was very nice, and you gave the general tips before the presentation how we want to prepare, and it was very nice tips. And I have a general question. So when we present about our result, do we need to mention about the number of animals in each slide, or do we mention about in the like that? Oh,
putting the N. Yeah, that's a good idea. A lot of people like to see that, so you're right. So that's a good critique of mine. I'm in trouble. Thank you. This was kind of an old presentation, but yes, having the N, having the dot plots instead of the bar graphs, kind of modernizing it so that the data is more transparent. Yes, that's a good point. Thank you. And a lot of stuff is personal style. So each lab's going to have their own style, you know, figures. Another point is to make it big. So lately I've been seeing, so we just had med student grand rounds, and a lot of people like to put the figures small and then text big, and it was really hard to read the figures. Some would put small text bunched in the middle of the slide, and the rest was a lot of white space. So really kind of make it as big as you can, but have white space as well. Okay, so make sure that people in the back of the room could see. And make sure you're using good contrast too. If you're putting, you know, I'm red-green colorblind, so a lot of the red-green immunofluorescent images are difficult for me. So don't put blue on a black background, you know, just, you know, keep that in mind, have a really good contrast. So good points. Any other points or things to address? All right. Stacy is going to make this available on, I believe, YouTube.
Yes. So if you would like to send me the slides, I can email those to everybody along with a link to where it'll be posted on YouTube. It may take me a couple of days to post that, but yeah, I'll let everybody who registered know how to find that.
Okay.
Okay, I don't see any other questions in chat. So thank you so much, Dr. Mitchell. I appreciate it. And everybody remember to sign up for the webinars we're doing every other week. Thank you.
Yeah,
Thanks,
sure.
everyone.
Thank you. Thank you.
Thank you.